Hello and welcome to today's webinar. I'd like to go over the agenda for what we're going to cover. So firstly, we'll start with myself going over an introduction to AB-SciX and the product portfolio. Then I'll hand to Richard Sleeman from Spec Analytical, who will go over the use of AB-SciX triple quadrupole instruments for trace detection of explosives and illicit substances for forensic purposes. So first of all, we'll start with an introduction to AB-SciX. AB-SciX is a part of Danaher Corporation, which is a large corporation of multiple companies in different market segments. Danaher is quite a unique company that acquires lots of different companies but keeps their brand. You can see here from the market segments that they play in quite a diverse area of environmental, testing and measurement, in the dental area, life science and diagnostics, and industrial technologies. AB-SciX sits within the life science and diagnostics area and has sister companies such as Beckman Coulter, Leica Microsystems, Radiometer, and Molecular Devices. AB-SciX is a global leader in mass spectrometry with over 20 years experience of manufacturing mass spectrometry devices, and also has service, support and software portfolios to drive customers' business. AB-SciX interacts with their customers by really listening to what the customers' needs are and building a partnership with the customers and really helping them to answer the questions that they need to really drive their research or their testing and to basically improve the quality of life. AB-SciX plays in a number of different market areas within analytical science. Firstly, pharmaceutical area like drug discovery and development, then in the clinical research and forensic toxicology area, then moving into more research within the universities, talking about biomarker discovery or proteomics research, and then in the food safety and environmental testing area or water testing. Back in 2011, this was AB-SciX's instrument portfolio, and since then we've worked very hard to update and expand our portfolio of instruments and technologies, and in 2012 introduced a number of new products in terms of the 4500 series of triple quadrupole and Q-trap instrument and the 6500 triple quadrupole and Q-trap instrument. Also recently, with the acquisition of Exigent, we now have our own chromatography systems that we can work at standard flow, but we also have microflow and nanoflow instruments. Moving into the accurate mass area, we have a range of accurate mass instruments now that are QTOF instruments in terms of the 4600 and the 5600, which can do much more advanced testing in terms of screening applications and also discovery and identification applications. So a conclusion of AB-SciX and our portfolio of instruments, we really have a range of platforms that can meet the sensitivity and compound identification needs. We have the unique Q-trap technology, which gives an extra level of confidence when doing compound identification. Within the toxicology, forensic toxicology area, we have targeted and untargeted screening workflows that provide excellent complementary approaches. And our instruments have been applied in multiple forensic laboratories across the globe to do various different types of forensic drug and explosive detection. So that concludes the introduction part about AB-SciX, and now I'd like to hand over to Richard to really discuss a bit more of the use of the AB-SciX instruments for the trace detection. Richard?
Thank you very much, Russell. My name is Richard Sleeman. I'm the Scientific Director of Spec Analytical. We're a company that was established in Bristol, UK 20 years ago. We provide forensic services to the police and law enforcement agencies. We've been involved giving expert evidence in many thousands of court cases. We're ISO 17025 accredited for our trace work as well as the identification of bulk drugs. And we also build sample introduction systems for mass spectrometers, and I want to talk about one of our sample introduction systems which couples with an AB-SciX triple quadrupole mass spectrometer. First of all, trace detection is invaluable to law enforcement agencies because they have a requirement to associate particular people with certain criminal activities. And one way in which they can do that is to identify involvement with specified illicit substances such as drugs of abuse. Now, the police don't need to know exactly what is on an item, such as clothing item or telephone, or they want to know whether a particular compound is present. So what is required is the fast identification of targeted illicit materials. Now, the conventional way of doing that is to, for example, shake a garment over a cover on the table, collect together the debris that comes off it, dissolve that in a solvent, reduce the volume of the solvent and filter it, and introduce it via syringe into a GCMS or an LCMS device. Now, obviously, that's a very time-consuming process, and we wanted to try and find a way which would minimize the sample handling and would not lose the evidence of where the traces may have been present. So our approach was to use thermal desorption tandem mass spectrometry to investigate the analytes of interest. Now, most of the drugs and explosives can be analysed with quite high selectivity using tandem mass spectrometry alone, without the need for chromatography. Now, chromatography is obviously time-consuming, so if you can eliminate chromatography, you can get much more rapid analyses and therefore perform more of them. So our approach was to heat samples up, collect the vapor that desorbed from them, and then pull those vapors into a mass spectrometer for direct analysis. And that gets rid of the need for glassware, solvents and chromatography, and saves time and obviously saves money. This is a schematic of how our thermal desorbers work. Basically, there's two heated plates between which the item of interest is placed, and that could be a paper swab or a toothpick or a banknote directly between the two plates. The drugs or explosives evaporate from the surface of the sample and are entrained in a stream of air, which is delivered to the mass spectrometer for analysis. One advantage of this approach is that the analysis results are available in real time, as opposed to perhaps 30 minutes for a chromatographic approach. And that not only does that mean that that more samples can be analysed in the time available, but it means that information concerning, for example, contamination of your working environment is immediately available, and steps can be taken to act on that. Another advantage is that very often in trace analysis, the police or law enforcement agencies are criticised for not being fully trained to collect trace exhibits and having caused the contamination themselves. Now, one thing that you're absolutely certain that they have handled is the exhibit bag. So testing the exhibit bag, even before it's opened, can be a useful guide to suggest whether or not the police were free from contamination. Now, our Sentinel system is a combination of the AB-SCIEX triple quadrupole mass spectrometer with our thermal desorption front end. It also incorporates a nitrogen generator and software which enables the system to be used by non-specialist operators. It provides a simple red light, green light display, so you don't need to be a chemist to interpret the results. We also provide swabbing kits which CSIs can take out to collect their own evidence and return to the lab for analysis. We have a field transportable Sentinel equipment in a van, which is used by the police to investigate large venues, concert halls, stadia, for example, in order to identify explosives threats or other threats. The requirements for such a system is it must have high sensitivity to ensure that there's a high probability of detecting threats should they be present. And also high selectivity is required because you don't want to get it wrong, because the consequences of finding, for example, explosives in a stadium may be that you have to evacuate the stadium. So you have to be pretty certain that your identification of the material is correct before you make such an important decision. The system has also been used in Canada by the Canadian Border Services Agency, and they've published a publication in the Journal of Forensic Science which uses the Sentinel system as the gold standard against which they compare the explosives detectors that are deployed in airports. The Sentinel system is used to rapidly investigate the efficacy of the other approaches that they use, and they say that the direct analysis transformed the traditional day-long quantification by solvent extraction to a matter of minutes. One common requirement is for money to be analysed for traces of drugs. Now, it's well known that money in general circulation is often contaminated with drugs of abuse, so it's important to be able to differentiate seized money, which is believed to be associated with drugs activities, from money from general circulation. So we have the largest database in the world of background contamination. Hundreds of thousands of notes have been analysed in our lab over the years, and we have conducted work with the universities of Bristol and the University of Edinburgh to differentiate statistically the likelihood of money from a seizure being contaminated to an unusually high degree with a particular drug. And in order to do that, it's very important that to get good statistics you need lots of samples, so a rapid technique is invaluable. This is a case study where some money was analysed, but before we could get to the money, a warrant was required because the police believed that a particular industrial premises was being used for nefarious activities involving drugs. They couldn't get a warrant, but one of the CSIs used one of our brush swab kits to take a sample from the vent of the industrial premises. That sample was returned to the lab, and when it was analysed, it was discovered that it was contaminated with amphetamine. So that was sufficient evidence to get a warrant, and the search of the premises revealed a number of counting machines and a very large amount of cash. And when that was brought back to the lab and analysed, it was discovered that that was also contaminated with amphetamine. That resulted in a high-profile court case, and over £5 million worth of cash was seized, and multiple convictions resulted with very long prison sentences. So to summarise, we've tried to eliminate the need for sample preparation for the analysis of targeted trace compounds. We've coupled a thermal desorber with an AB-SciEx triple quadrupole mass spectrometer. That's more efficient than conventional methods using solvents. The location of traces is not destroyed during the testing, and the high sensitivity and selectivity delivered by the triple quadrupole device is highly important to minimize false alarms and have a high probability of identifying threats. Thank you very much.